Without further ado, I think we're just on time for our first speaker today. So our first speaker is Dr. Aondoyima Ioratim-Uba, who is a postdoctoral researcher in the Max Planck Institute for Dynamics and Self-Organization in Göttingen in Germany. So he first obtained a BSc in physics and then a master's degree in mathematical and theoretical physics at the University of Oxford. He earned a PhD from the University of Bristol, and his thesis title is Cells and Tissues as Active Matter. So, Jules.
Okay, so you have to go for 35 minutes plus 5 questions.
Yeah, that sounds good. Thank you so much to the organizers for having me. Today I'm going to talk a little bit about some work that I did during my PhD and kind of tell the story of how physics is relevant in biology. So to begin with, living systems are really complex. I kind of picked a handful of nice movies and pictures for this talk to kind of, so that we can see visually what's going on. A lot of, my bad, this doesn't seem to be working. Oh, there we go. A lot of organisms start life as a single cell, and they become something way more complex. And throughout that process, you have so many different things happening. You have cells dividing, cells sorting into different types and different categories based on where they are spatially. You have cells dying, cells sort of being extruded from where they are. You have sort of cells breaking symmetry, taking on asymmetric shapes. You have a lot of physical forces involved. Cells are sticking to each other, surface tension plays a role. And this is just the tip of the iceberg. And on top of this, there's also a lot of chemistry, there's also a lot of biology happening. But yeah, we're hopefully going to learn a bit more about the physics. So to start with, I'm just going to show, so this is really cool. This takes a while to load up, but this is a fruit fly embryo, a Drosophila embryo. And what you can see sort of glowing silver, the quality is not amazing, but it's the chromosomes inside the nucleus of each cell. And you can see the cells dividing, so you can literally see one cell becomes 2, becomes 4, etc. So this happens in the first 3 hours of development, and we're going to end up with about 6,000 cells. So let it go for a little bit longer. It's remarkable. You can literally see cell division. So yeah, this happens for a while, and then we get to before we get to the biology, I'll explain a little bit how maybe I went from that process to getting here. So I was born in Nigeria. I was born in Jos. It's a city with really cool rock formations. It's very mountainous. And then when I was 8 years old, I moved to Manchester. Spent a lot of time in Cuban Park. It's very nice. And while I was there, while I was in school, my teacher lent me a book by Jim Al-Khalili. And this book really got me obsessed with physics. And so after that, I went on to do physics at Oxford, did a PhD at Bristol, and then finishing my postdoc at the Max Planck Institute in Göttingen. Göttingen is really nice, where the Max Planck campus is. It's a lovely, lovely place. But yeah, so that's kind of how I got here. But yeah, so if we're thinking about going back to the Drosophila embryo, we had started with a single cell, those cells divided. That happens in about 3 hours till we have about 6,000 cells on the surface, way more inside. At this point, all the cells are more or less the same. Nothing that special is happening. We've just kind of had this thing grow. But at this point, and I think this is the point of development that I'm going to sort of zoom in on, we see some incredible stuff. Hopefully this video works. Oof. Yeah. It's not a big deal. There's a diagram. So basically, I'll explain what is happening. This actually is really, maybe if you can remember that link or ask me, I'd recommend watching this because it's really interesting. Essentially, what you see is this sort of mostly uniform oval of cells basically folds itself over, and you have a lot of different things happening. You have the tissue compressing in some places, you have the tissue stretching in some places, cells are differentiating, and sort of this is the process that sets up the body plan. So basically arrange the cells into where they're going to be in the body later on. So yeah, how does nature control the desired outcome? Because there are lots of moving parts, like millions and millions of different things happening at once. So yeah, in that top corner, brilliant, laser pointer. This is sort of what you were supposed to see. You were supposed to see an animation of this part of the embryo stretching out. And as to how that is coordinated, we have semantics biologists have been, on this for a long time before physicists came into the picture. And we know that spatial patterns of gene expression. So this picture here is certain genes have been made to be fluorescent when they're being expressed. And so we have a stripe pattern of two different types of gene being expressed in the embryo. I'm not going to get into too much into biological detail. I'm sure many of you know better than me. Yeah, so for example, this structure here, the cephalic furrow, it's basically determines where the head of the fly is going to be. And this is correlated with the expression of certain genes. And similarly, this process of tissue elongation, germ band extension, is associated with also patterns of gene expression. And so again, you see stripe patterns of gene expression. So this happens in the Drosophila embryo. It's kind of well studied that these patterns of gene expression really do play a role in shaping the embryo and determining its structure. But it's not the whole story. So I'm going to zoom in on germ band extension. And for the rest of this talk, really, I'm going to focus on what happens during germ band extension, which is that the tissue elongates. It gets longer along one axis and it gets narrower. So you can see here, sort of longer horizontally and narrower vertically. It's a fundamental process in development, so it happens in so many different organisms. It happens in, zebrafish, sort of amoeba on the bottom of the ocean, and frogs, and dozens. And it also happens in the chick primitive streak. So this is kind of what I'm going to focus on, because I will talk about a model that is relevant to convergence extension in the chick primitive streak. So it's a patch of tissue that elongates and narrows. So yeah, my take here is that gene expression doesn't fully explain convergence extension, partly because the mechanisms can be different in different organisms. And so those kind of patterns of gene expression we saw in the Drosophila, these stripe patterns of gene expression, are not really present in the same way in chick embryos. Instead, what seems more relevant, so what you're seeing here is a patch of tissue. You have a lot of cells, and the green here is the most relevant. It's showing the presence of actomycin, which is a complex made up of molecular motors and proteins. And the molecular motors are able to generate force inside of the tissue. And in chick embryos, we see long chains of these, of this actomycin complex. And essentially, these chains are under tension. And so the idea is that the tension in the chains contracts the tissue, and you get something, you get a tissue that, narrows due to the forces at the connections between cells. So if we zoom in a little bit onto the tissue, it kind of looks something like this. Each cell is basically a polygon, and it's connected to its neighbors by an edge. And what you're seeing kind of in this microscope picture is that the green here is localized. The molecular motors are localized to the cell-cell junctions. And yeah, just to give a clearer picture of what actomycin is, it's made up of two different things. It's made up of motor proteins and motor proteins and protein filaments, so long proteins. And basically what happens is that the motor proteins generate forces to either stretch or contract the filament proteins. And so we kind of have this picture where the force at cell-cell junctions, the tension really plays a role in what's happening in the tissue. One thing that is relevant, I'll just briefly mention it, is that the tension basically causes junctions to collapse. So if you look at this picture in the upper corner, what's happening is that the tissue is narrowing, and it's narrowing in the direction that's perpendicular to the direction of the myosin chains. So we have the chains going horizontally, and because of the tension, the chains are sort of collapsing in. And if you zoom in close enough, what you see is basically cells changing places because of that. And this is kind of a process that drives the elongation of the tissue. So yeah, I say that gene expression is not the full story because tissues are sensitive to forces. So if you apply force to a cell, for example, that force can cause changes in the cell nucleus, changes in the tension in the cell nucleus, and also changes in gene transcription as well. And in general, forces can change biochemical pathways. So if you apply force to a cell or a tissue, you can actually change its phenotype. You can change a lot of its chemical characteristics. And so what I'm kind of going to focus on in the rest of this talk is, well, can this process of convergence extension, can this process of tissue changing shape by narrowing and elongating, can it be triggered and sustained by such a feedback mechanism? And so instead of just patterns of gene expression, which happens in fruit flies, in other, especially bigger organisms, that mechanism might be less relevant. And so I'm going to think about, okay, well, maybe if I pull on the tissue, what if it generates forces to produce the desired extension? And in particular, actually, I'm going to talk about how the tissue generates forces that oppose the force that you apply to it. And so if I kind of pull on the tissue this way, its response is going to be to go the opposite way. In the same way that if someone were to pull on your arms, you would be capable of pulling back in. And they can do this because of the molecular motors, because the myosin motors can produce force, because they consume energy. And so this only happens because tissue is living. And therefore it's out of equilibrium. Okay, so I'm going to start with what happens in just a single junction. So we have a tissue, it's made up of all of these cells that are connected by junctions. The junctions have motor proteins localized on them. And so they have myosin motor proteins, which are bound to the junction. And then there's some myosin which is not bound, but can become bound. And so there's kind of a process where the junction is constantly exchanging myosin with what are the cells in its environment. And this model basically says that if we increase the tension in the junction, so if we pull on the junction, what that's going to do is it's going to decrease the rate at which myosin unbinds from the junction. And so we have this kind of process where myosin is binding and unbinding, but suddenly you apply tension and all of the myosin on the junction is like, no, I want to stay, I'm not going anywhere. But that increases the tension because myosin produces force. And so if you're kind of basically decreasing the myosin that's leaving the junction, you're increasing the force. But then that's a feedback loop because that's the process that decreases the unbinding of myosin. And so you have this kind of feedback. The sort of the relevant thing about the model is that the junction is viscoelastic, which basically means, I'm not going to spend too much time on it, but it means it can change its rest length. And so you have a junction with some length and then some length that it wants to be. But that length that it wants to be can actually change. That's kind of maybe the most relevant part about the viscoelastic side of things. But really, I want to focus on this mechanochemical feedback. And so which increasing the tension, that tension is basically allowing more myosin to stay on the junction, which increases the tension. And so what do you think might happen in that situation? Well, this graph shows how the junction length changes over time. And so these are simulations that show that essentially if you apply tension to the junction, it will shrink and it can shrink to zero, which is what these 3 points are showing. The different lines are just junctions of different stiffnesses, more or less. And equivalently, you can see the concentration of myosin on the junction increasing concurrently with the junction collapsing. And so that's kind of good. This really simple model displays behavior that we see in biological tissue. Like I mentioned before, I think it's worth going back. Like I mentioned before here, we see in tissues that are undergoing convergence extension, we really do see these junctions collapsing to zero and extending out in the perpendicular direction.
So he was just on the plot with the L against
Yeah.
So it's L divided by, I guess it's resting length.
Yeah. Actually, I can't. It might just be out in this case.
Yeah.
Just like the raw tension length.
It was just set to 1 for like simplicity.
Yeah. Oh yes. Yeah. I see what you mean. Yes, it is L. Yeah. That's 1 there. Yeah. Yeah. So we can see that this really simple model gives us some biologically relevant behavior. It gives us the junction collapse. So that's kind of hopeful. So can we describe the whole tissue? The answer is yes. What we need is to consider the fact that when we don't have a single junction, the orientation matters. So for the single junction, it doesn't really matter. All it is, it's just that. But then when you have a lot of them connected to each other, suddenly you care about, well, am I 90 degrees, am I 35? Am I, like, what angle am I pointing at? And so instead of a single concentration, we basically need a tensor. We need a tensor that describes that can tell us about the orientation and the intensity of myosin. So this quantity here, M, will tell us about for a given junction, J. So for example, junction 1 has a vector that's pointing in the sorry, I'm tripping over my words. This junction 1 is described by a vector L1 that's parallel to it. And it has a concentration of myosin M1. And in this diagram, we're sort of saying that the darker color is low and the brighter color is a high concentration, just to kind of illustrate what's going on in the tissue. So great, we kind of have this quantity, but what does it actually mean? So first of all, what I mean by this kind of, I don't know, the notation is always a bit different, but this is essentially what an outer product. This is if I have a vector AB, so we're in plane, all L cross L means, so the outer product means is that we get a matrix that looks like this, whose entries are A squared, B squared, and AB. And so this, so I'm currently defining this only for a single cell. So what does the myosin tensor look like for a cell that has equal junction lengths, because junction length as well as orientation matters, equal junction length and equal concentration of actomyosin on the junctions? We get the identity, which is nice. So this tensor encodes some symmetry. If you have a junction with not equal concentrations of myosin, so only 2 of the junctions here have any myosin on them, we get something different. In particular, it's not the identity and the diagonal components not the same as each other. If we have a junction if we have a cell whose junctions only have myosin the vertical junctions have myosin, we have something where only the YY component of the tensor has a value, and similarly, if we flip that by 90 degrees, this is what the actomyosin tensor looks like. So hopefully that should give you, and I think that'll come back a bit later when I talk about what the model actually does, but hopefully that should give you some intuition for how this tensor kind of encodes distribution of actomyosin. Okay, so what I'm going to talk about is a continuum model. So chick embryos are actually very big. And so the picture of the cells I showed you before was very zoomed in. But if you look at them kind of a little bit more zoomed out, it's actually very hard to make out the individual cells. And so that kind of motivates the idea of a continuum model. So instead of looking at each individual cell, we're going to, for each position, take an average over lots of cells. And so we can define the continuum actomyosin tensor that for each spatial point will give you so in the continuum description for each point x, y, I will have a matrix like this telling me how the atomizing is distributed at that point. And so this is kind of a summary of it. We have a tissue, the junctions have atomizing on them. The dynamics of the tissue is going to be described by the atomizing tensor. We also need a passive stress tensor, which is going to satisfy compressible viscoelastic dynamics. And we have a velocity field that tells us basically how the tissue is flowing. And so the model has three equations. The first is an equation for the velocity field. And the second is two equations, one for the passive stress and one for the atomizing tensor. This quantity beta is the activity, and it basically tells us how much force the myosin molecular motors can produce. And M0 is a base level of myosin. So in the model, the junctions have just sort of a base level of myosin that defines the default configuration. So this equation, if you remember what the single junction equation looks like, it's very similar. On the left, I'm not going to spend too much time here, but the left-hand side is basically the co-rotational derivative of the tensor. And what that means is that it's a quantity that lets you describe the tissue in a way that doesn't depend on the coordinates that you use. So if I change the coordinate system I'm using, this thing stays the same, which is what we want. Tau m is the myosin binding time. This is the most important piece of the model. It's basically the tensor version of what we saw in the single junction version. And the last term is diffusion. Describes diffusion of atomizing in the tissue. For the compressible viscoelastic dynamics, we basically have the passive stress tensor, the trace and the off-diagonal components are going to have independent dynamics, and we can basically take a linear combination of that. So tau v is the viscous relaxation time, and then these two quantities, e to p and e to s, describe bulk viscosity and shear viscosity in the tissue, and then we have strain rate. But this is, I guess, quite fun, but I think really what I want to show you what the model actually does. And I'd love to answer any more questions about any details of the model. Okay, so the first thing that we see is that if we take this tissue and apply forces to it, so if we compress it vertically and stretch it horizontally, it's going to do the exact opposite thing that you did to it. It's going to compress inwards and, oh my bad, I'm so sorry, these arrows are, that's really annoying. These arrows are the wrong way around, which kind of defeats the whole point of what I'm trying to say. But there'll be later on versions of this with the correct arrows. So ignore those arrows. What this is supposed to do is the exact opposite of, and I think you can make out the vectors. You can see that the tissue is flowing inwards here and it's going up here. And so yeah, the tissue is doing the exact opposite thing that you do to it. And what's really interesting is that interesting, the labels are not. Some of the, anyway, it doesn't really matter. This is the XX component. So this here is referring to, this is telling me how this component of the tensor is changing in space throughout the tissue. So this plot is showing the X and Y. It's basically showing spatial variation of atomizing and the passive stress in the tissue. Really, we want to focus up here. And so what we can see is that this tissue has a very high XX component throughout and a very low YY component throughout. And if you can remember what I showed you with the single cell, you might be able to start to figure out what's going on. And so for a single cell, a very high XX and a very low YY component would correspond to the atomizing being sort of parallel to the horizontal direction. So we can dig a little bit deeper as to what's going on here. And these equations have a very nice keep going. These equations have a very nice solution. If you set the time and spatial derivatives to 0, the components of the atomizing tensor, so the mxx and myy, essentially you can assume the off diagonals is 0 for everything that I'm going to say, maybe up until the very last slide, the off diagonals is 0. And so we can think of, okay, what happens if we just assume that everything is uniform in space and uniform in time, just as a really simple way to get a feel for how this model works. What happens? Well, what we see is that if, as a function of activity, so basically as a function of the strength of the molecular motors, we see that initially the atomizing concentration stays at the base level, and then at some value of activity, there's a bifurcation. So it can either be above or below the base level. And what we're seeing in this convergence extension flow, so where the tissue is flowing inwards and extending out, extending up and down, we see that the XX component of the atomizing tensor goes above the base level, and the YY component goes below the base level. And this is basically telling us that there's this polarization. We have a preferential direction which atomizing wants to be concentrated along, and it is essentially the horizontal direction. And so that's quite nice. This sort of so far is agreeing with the picture that tension chains along a particular direction can give you convergence extension, can give you this kind of flow. We can also do simulations. And so if we do something a bit more complicated than just everything is uniform in space and time, the first thing that we see, okay, so let me explain what this diagram is. On the y-axis, we have the strain rate, and that's basically telling you information about how the tissue is deforming, how it's changing shape. And on the x-axis, we have the applied stress. What you want to look out for here is that when the strain rate goes negative, so any parts of the curve below the 0 line, that's corresponding to this kind of behavior, where the tissue is doing the opposite thing to the force that you applied to it. And what's nice is that the simulations also have this happening at the same threshold activity. And so basically, if you look below 0.5, so for a value of activity of 0 and 0.3, the strain rate is always positive. And so that's kind of behaving like a normal material. It's behaving like a material where you pull it and it goes in the direction that you pull it in. But then above 0.5, you start to see a negative strain rate. And this is a signature that the material it basically has opposite behavior. Okay. So applied stress is a mechanical signal in that if you change, these forces don't have to be very large. You can basically apply a tiny force to induce the tissue to flow in a certain way. Really annoyed about those arrows, but this is what I mean. So I think these are all correct, yes. So you can see here, if I push the tissue along the X direction and extend it along the Y direction, it's going to do the opposite thing. Similarly, so these are all data from simulations showing the velocity field of the tissue. If I pull the tissue along all directions, it's going to compress itself. And if I try to compress the tissue, it's going to extend. So this behavior only happens above the threshold activity. And well, it depends on the fact that the tissue is living. And so a passive material couldn't do this. A piece of elastic, for example, couldn't do this. So, and again, the behavior for the different boundary conditions that I apply basically selects which of these branches that the components of the atomizing tensor lie on. And so this situation, you have the YY component being above the threshold and the XX below. These final two situations actually have both components of the tensor picking the same branch. So yeah, this model kind of, this bifurcation picture basically is enough to figure out how the tissue should be, how the tissue is polarized. Okay, so apart from convergence extension, the mechanical feedback mechanism also gives rise to other behavior in the tissue. And so convergence extension is what we saw is when the tissue is going in one direction and going out in the other. But we also have other things happening. We have oscillations and sort of really slow, complex facial patterns. So there's kind of a lot to explore here in terms of what might be relevant to other biological processes, or just relevant to maybe designing active materials. So here I'm changing the tissue properties. I'm changing the viscous relaxation time, and I'm changing the myosin binding time. I don't know if this is a chance to press play. So that's, yeah, so that's working. Essentially, what you can see here are the diagonal components of the actin-myosin tensor and the two components of the passive stress tensor. I think the kind of more intuitive one is this picture. So this is showing how the tissue is flowing. And you can basically see, I kind of put these here because I like the way that they look. These very pleasing patterns. And you can also get oscillations as well in the system. And again, I just think these are quite pretty.
I think the audience might have to explain how you get that, because it's not the points aren't the cells, it's sort of you fix points in space. Maybe just
yeah, absolutely. So what this will make sense, I promise. So yeah, how I get that is going back to this idea that the tissue is really big. And so if you're looking at it from a sort of overall perspective, it's very hard to make out individual cells. So what we can do is we can basically pick a point and say that the velocity at this point, the value of the actin-myosin tensor, the stress, is all an average of the cells in this point. And so in all of the diagrams that I'm showing, each arrow, for example, represents an average of what the cells around that point are doing. And so yeah, this picture kind of only makes sense if you look at it from afar. It's kind of giving you the overall flow of the tissue. Was that, yeah, nice. But yeah, we're essentially at the end of my talk. So I just kind of wanted to show these other two behaviors to say that mechanical feedback is, yeah, really interesting. It gives rise to a lot of different complex behaviors. Okay, so yeah, biological development was really complex. There's a lot going on, but it manages to be robust. Most organisms kind of come out the same, with the same number of eyes, the same number of limbs, the same shape of limbs, despite size. And so that's really interesting that such a complex and messy process can be that robust. We can explain some of it with biochemical signaling. So with the fruit fly embryo, we saw that patterns of gene expression can get you the desired shape. You can basically correlate patterns of gene expression with structure in the embryo. But if we're looking at other organisms or want to have a fuller description of what's going on in the embryo, we need a combination of mechanical and biochemical signaling. And so the example I showed is one where you increase junction tension and you decrease myosin unbinding, and that kind of generates a feedback loop that continues to increase the tension in cell-cell junctions. So this mechanism gives rise to convergence extension triggered by a small applied force. So if I hold the tissue slightly, it's going to respond by going in the opposite way and then elongating in the perpendicular direction. That's the main thing I want you to take away from this talk, which is that knowing the arrow is not the right way. So these should be going up. I'm very sorry about that. But yeah, thank you very much. Thank you.
So yeah, thanks, James, for that very enlightening talk. So we have about 10 minutes for questions. So yeah, any questions from the audience or on Zoom? No, I'm happy to pick up some questions and ask people.
Yeah, so I know nothing about this area, so this has been, my questions will be very low level, I'm sure. But
The best questions.
Sorry?
The best questions.
The hard.
So I suppose honestly, this is, first of all, what kind of forces are we talking about?
Kind of, so I'm mostly talking about tension when I'm talking about rejection. So really like pulling or compressing.
Order of magnitude.
Oh, I see what you mean. That's a very good question. I actually I don't have a good answer for you.
Right.
But I might be able to dig something up.
Human level.
No, they're much smaller.
Yeah.
Yeah, I don't want to say them because it's probably gonna be wrong, but I can we can chat after and I can give you a number. Sorry to the audience.
Yeah,
That's actually a really good
I
question.
think you've actually answered on
Yeah.
very general. And then, so I'm assuming then that when these interactions happen, they also interact in the cells, and then they're causing tension on those, and
Exactly.
it's like a chain reaction.
Yeah,
Right,
yeah.
got it, and that's what that diagram is. Yeah. Wonderful, thank you.
Nice. So we have a question from the Zoom. So one assumption appears to be that cells are healthy. Is it possible using your model to distinguish unhealthy cells from healthy?
That's good. This model currently, maybe not, but unhealthy cells do have like physical signatures. So there could be a way to incorporate a kind of measure of what an unhealthy cell is based on, I don't know, maybe you could, unhealthy cells can not produce enough tension. So maybe for some of your tissue, you can have I don't know, you can have the level of tension capped, for example, and that might capture unhealthy behavior. So in this model, as it is, no, but actually there might be a few simple ways that you can incorporate that kind of behavior.
Yeah, so I have a question actually, just a bit sort of tongue-in-cheek Feynman-like question. So you I know the video didn't show, but you described quite elegantly this. So the model assumes a sort of flat 2D sheet, but really in practice this beautiful movie of folding tissue in 3 dimensions and twists and so on.
Yeah.
So is it possible sort of extending the model to look at sort of twisting? So instead of pulling, you have like twists, rotational stresses, and also if you sort of have it on the manifold where you have curvature.
Yeah.
I mean, what would that look like?
Yeah. So actually there have been people that have attempted that. I think there are some models that look at actually, the surface, a curve, these behaviors on a curved surface. And essentially you get very similar outcomes. So it's kind of nice because looking at it in 2D, although it's a simplification, it gives you the essence of the problem. Because we're not going to be describing biology anyway, like accurately with such a physical model. Like there are millions of things happening. And so we're like, okay, what's the most important ingredient? And in this case, the feedback mechanism has a way bigger sort of impact than the curvature. But it would be interesting to see especially twisting as well. I think that's actually a really cool question because so far we're just basically applying forces that don't involve any twisting motion. So yeah, what happens if you take the tissue and try and twist it? That's interesting. I imagine it might, I don't know, try and twist in the opposite direction or generate maybe opposing spirals. But yeah, that's just pure speculation.
So I guess maybe yeah.
Yeah, I was wondering about the range of potential like tension space across different species. So I guess all of your models are done on the chick one. How does that compare to a zebrafish or some things from different temperatures where the tension is going to vary?
That's a good question. So this model, although I focused on chick, it's sort of, there's nothing in the model that says it has to be a chick embryo. All it's saying is that feedback mechanism exists. So I guess it depends whether that particular organism has the mechanism or not. And we know that convergence extension happens actually in different ways and by different mechanisms in different organisms. So yeah, it really depends on what the specific is present. But I think ideally it should apply to a wider range. Yeah. Yeah, exactly. Yeah.
Yeah, so a couple of interesting things then on that, like how common is a kind of biological? Is it something that happens to what proportion of species?
Yeah, so by this do you mean the feedback mechanism or convergence extension?
Yeah.
So yeah, so well, I guess I'll answer both. The feedback mechanism, it seems quite common. It seems that maybe not this specific mechanism, but just in general, I think pretty much all biological organisms have some type of feedback because they're mechanosensitive. So if you apply force to a tissue or a protein, I think it's capable of responding and changing. So I think I think that's probably kind of the underpinning thing of living matter, especially if it exists in 3D space. That actually there's constant feedback between mechanical signaling and biochemical signaling. So yeah, I think it happens pretty much everywhere. And convergence extension also does as well. I think basically how tissue changes shape is that's one of the ways that it does it, by narrowing and elongating.
And is that throughout life, or is it
through usually development? I don't actually know if it happens maybe during wound healing. So when the tissue is damaged and then it has to flow, maybe you might get some of the things, but this is mostly relevant to development.
So maybe just kind of going beyond science. So you're here at the Black Heroes of Mathematics conference, and so we're celebrating Albert Cox's 100 years since the first Black PhD in mathematics. So what does it mean to be here, and what are your thoughts about I guess this sort of 100 years since the first Black PhD, or you can also say about your future direction and what you would like to contribute.
I mean, it's really special to be here. 100 years seems short. Seems really, really short. So that's kind of strange. But yeah, I mean, it's I don't know, it's kind of nice to see, so many Black mathematicians here, because I'm sure we're all used to maybe being the only one in wherever we are. So it's really comforting. And yeah, I don't know, I guess just it's nice to show it being done, to show examples. I think the more it is seen, the more it's normalized. You kind of, I guess, there's that weird psychological thing of not feeling you can do it until you see someone that looks like you do it, which might sound trivial, but it's actually kind of, it's one of the most important things. And so I think I'd just like to continue showing that it can be done.
I mean, that's a great note to end on. So let's thank Deems for a beautiful